Hello, middle 3. Let's complete lesson 1, which was called cell division. We took last video the mitosis division or the mitotic cell division. Today we will talk about the second type of cell division, which is called meiosis. Okay, meiosis occurs only in reproductive cells. Where is the reproductive cells? Reproductive cells are the cells present in reproductive organs, or the gonads, which are the testes in male and ovary in female. This is in man and animals. In plants, it's the anther in male and the ovary in female. Okay? These are the gonads, or the reproductive organs. طيب. What does the meiosis do? It occurs in the reproductive organ to produce something called gametes. What's the gametes? The gametes are the sex cells, the cells which are responsible for the reproduction, which are sperms in male animal or man and ova in female animal or man. Okay? And in plants, it's pollen grains in male plants, okay, and ova also in female plants. What's the importance of meiosis? It's important for the production of male and female gamete to complete the sexual reproduction. Okay? Let's start بقى the stages of meiosis. Meiosis takes place in two main stages. First meiotic division و second meiotic division. First meiotic و second meiotic. Okay. Before the first meiotic and second meiotic, there is the interphase stage, which is the same as in mitosis. In interphase, the cell prepares itself for undergoing the division and it duplicates its genetic material, okay, and undergoes certain processes. Let's start now the first meiotic division. It aims to reduce the chromosomal number to the half. يعني ايه؟ reproductive cells اللي هي موجودة in the reproductive organ contains diploid number of chromosomes اللي هي 2n. Okay? I need to form a gamete. A gamete contain haploid number أو n واحدة. So I need to reduce the number of chromosomes from 2n to n. So this is made by the first meiotic division. So it's called reduction division. Chromosomes are doubled once in the interphase that takes place before the beginning of meiotic division. Yes, as I said. Now let's start phase number 1 in the first meiotic division. We will call the phases of the first meiotic prophase, metaphase, anaphase, telophase as the same as in mitosis, but here we have to put 1. Prophase 1, metaphase 1, anaphase 1, telophase 1. Why? Because the meiosis have two stages. First meiotic و second division و second meiotic. So we need to say 1 after the name of each stage or 2 after the name of each stage, so you will know which stage you are talking about. Is it the first meiotic or the second meiotic? Okay? يبقى prophase 1, the chromatin reticulum intensify and appear in the form of chromosomes. Okay? The nuclear membrane disappear, the nucleolus disappear, and the spindle fibers start to form and extend between the two poles. Then the chromosomes are arranged in homologous pairs. This is the difference between the prophase 1 and the prophase in mitosis. In mitosis, the chromosomes were in singular pattern, يعني each chromosome is alone on its own, okay? Here in prophase 1, the chromosomes are arranged in pairs, okay? They stand in pairs. Each two homologous chromosomes stand near each other, okay? They are called a tetrad. Why a tetrad? Because they contain 4 chromatids. They contain 4 chromatids. What happens then? A new phenomena happens here in the meiosis, in the first meiotic stage, which is called the crossing over phenomena. What's the crossing over phenomena? It's a phenomena that occurs at the end of prophase 1. Okay, some parts of the 2 inner chromatids of each tetrad are exchanged to produce new genetic arrangement. As you see here in this picture, the 2 homologous chromosomes are standing near each other, okay? Then crossing over happens, the two inner chromatids exchange parts, okay? Exchange parts. This leads to or produces new genetic arrangement, which leads to variations in the genetic traits in the members of the same species. So that's why you and your brother and your sister are not the same, okay? You are similar in some things, but you are different in a lot of things. When the crossing over is finished, at the end of prophase 1, the two homologous chromosomes get away from each other. Yes, they were linked to exchange the part, then they stand near each other, but they move, the inner chromatids are away from each other, okay? Then each chromosome consists of two chromatids linked together by centromere, we know this. Then the spindle fibers are connected to the centromere of the chromosome. This is the end of the prophase 1. What's the importance of crossing over? As I said, it contributes in genes that carry the genetic traits, exchanging between the two homologous chromosomes, chromatids, and distributing them randomly in the gametes. This is an important factor for the variation of genetic traits among the members of the same species. As I said up when I was talking about the crossing over, this is why you are different from your brother or your sister. Let's now go to the next stage, which is the metaphase. In metaphase, M is middle. Yes, the chromosomes are arranged in the equator of the cell or in the middle of the cell. Here, the chromosomes are not arranged in singles. They are arranged in pairs, okay, in homologous pairs. Then anaphase 1, what happens in the anaphase 1? In this phase, the two homologous chromosomes start to move away from each other as the spindle fibers shrink. One of the two chromosomes migrates towards a cell pole and the other migrates toward the other cell pole. So each pole contains half the number of chromosomes of the parent cell. At this time, at this stage, yes, the chromosomes were divided into two halves. 23 in this cell and 23 in this cell, okay? In telophase 1, what happens? Yes, the spindle fibers start to disappear, the nuclear membrane start to form, the nucleolus start to form, and each cell contains half the number of, or the original number of chromosomes of the parent cell, okay? So at this stage, I have two cells, two new cells or two daughter cells containing half the number of chromosomes as the parent cell. Now let's go to the second meiotic division. In the second meiotic division, it aims here to increase the number of the produced cells from the first meiotic division. يعني I have two cells, okay, produced in the first meiotic division. I want to increase the number. I want, instead of 2, I want to have 4 gametes. So in this stage, or in the second meiotic division, I will do the same as in the mitosis division. I have 4 stages: prophase 2, metaphase 2, anaphase 2, telophase 2, okay? It's the same as in mitosis. What happens in mitosis? As we know, yes, mitosis have prophase, metaphase, anaphase, and telophase. The same in second meiotic division. Prophase 2, metaphase 2, anaphase 2, telophase 2. The same steps is done here. In prophase 2, the nucleolus disappear, the nuclear membrane disappear, the spindle fibers start to form, extending between the two poles, and the spindle fibers are attached to the centromere of the chromosomes. Here in prophase 2, the chromosomes are not arranged near each other, or are not arranged in pairs. They are not homologous chromosomes here. Each chromosome is on its own, okay, as in the mitosis. Then in metaphase 2, what happens? The chromosomes are arranged on the equator, or in the equator of the cell, or in the middle of the cell. Then in anaphase 2, what happens? The centrosome starts to split into 2 halves, and each one of the chromatid moves to one pole of the cell. طيب, how many chromosomes in metaphase 2 here were arranged in the equator? There are only 23 chromosomes. Remember, in the first meiotic division, I reduced the number from 46 to chromosome. From 46 to 23 chromosome. So here in the second meiotic division, I have 2 cells. Each of them contains 23 chromosomes. So in the metaphase, I have in the middle of the cell 23 chromosomes here and 23 chromosomes here. Then, as I said, in anaphase 2, the centromere splits into 2 halves and the 2 chromatids move away from each other, each to one pole of the cell. At the end, in telophase 2, what happens? The nuclear membrane starts to form, the nucleolus start to form, the spindle fibers start to disappear, and I have 4 cells, but not 2 cells, I have 4 cells because each cell divided into 2. Each one of these cells contains half the number of the parent cell that I started from the first meiotic division. Okay? يبقى I have here at the end of the second meiotic division 4 cells. Each one of them contains half the number of chromosomes as the parent cell. These cells are called gametes. As I said, in male, they are sperms or pollen grains in plants, and in female, they are called ova. They are gametes, okay? طيب, when the male gametes combine with the female gamete, the zygote is formed. طيب, the zygote contains how many chromosomes? 23 from the male gamete, 23 from the female gamete, so it contains 46 chromosomes. So here I got to make the number of chromosomes constant, remains constant in the cell of the members of the same species. Okay? طيب, let's talk now about an application of technological application about the cell division. In this part, we will talk about the cancer. طيب, what do you know about cancer? As I said in the first part of the lesson, that the cell takes 90% of its lifetime in the interphase stage. طيب, what does it do in the interphase stage? It prepares itself for the cell division by doing certain biological processes, and it duplicates its DNA. But only 10% of its life cycle it divides, or it undergoes the mitosis or meiosis. So the division is not all the time, okay? It's only part, very, very small part of the life cycle. طيب, what if the cell contains something abnormal, or it undergoes abnormality, and then it started to divide all the time? It undergoes uncontrolled division. What happens here? This is cancer, okay? This is what's called cancer. So cancer occurs when the body cells are divided continuously without controlling. This results in the formation of mass, as you see, mass of cells here. This mass of cells is called tumor. So this is what cancer means. طيب, a lot of scientists started to search for a lot of methods to detect cancer cells and to treat cancer. طيب, one of the methods to detect cancer cells was developed by the Egyptian scientist Dr. Mustafa al-Sayed. Dr. Mustafa al-Sayed detected a very new and perfect way to detect cancer cells, which is called nanotechnology. This nanotechnology used nanomolecules of gold molecule. What's meant by nano? Nano means very, very, very small molecules. Okay? It's measured by nanometer unit. But again, what does the nanomolecule of gold do? The nanomolecule of gold are used to help me see the cancer cells, or help me track the cancer cells in the microscope. طيب, again, the cancer cell produce a lot of secretions, okay? And some proteins attached to these secretions. So Dr. Mustafa brought these proteins and loaded them by gold molecule, nanomolecules of gold. Then he injected these proteins with the gold molecule in the patient blood. What happens? The protein with the gold molecule move in the blood of the patient until it reaches the cancer cells. So what happens when the protein sees the cancer cells? It attaches to the secretions on this cell. So when I look in the microscope, what will I see? I will see the golden molecule near or on the cancer cell. So I will know the place of the cancer, which cells exactly contains cancer and which cells are normal. So when I treat the cancer, I can treat it يعني in a good way without harming the normal cells. Okay. طيب. This is the detecting. Then what about the treatment? The treatment was by focusing laser with a certain degree to the gold molecule. But what happens when I focus the laser on the gold molecules? The gold molecule absorbs the light rays which are coming from the laser and then change them to heat. What happened when I have a lot of heat inside the cancer cell? The cancer cell will burn and kill itself. Okay? So in this way of treatment, what happens? The gold molecule are on the cancer cell by the proteins. Okay, the proteins attach to the secretions and the proteins carry gold molecule. So the gold molecules now are on the cancer cell. When I focus the laser, I focus it on the gold molecules. Okay, so the gold molecule absorb the laser and change the light molecule into heat. What happens then? The cancer cell starts to burn and it will be killed. Another way of treating cancer was by a lot of scientists, some scientists developed microscopic bombs, smart microscopic bombs. What these bombs do? These bombs penetrate the cancer cells and explode them from inside. This technique was experimented on some mice. These mice, of course, have cancer. So when they injected them with the smart microscopic bomb, the cancer cells exploded. The mice which were injected with the treatment or with the microscopic bombs could live for 300 days, while the mice who didn't receive the treatment only lived for 43 days. Okay, so this treatment showed that it was effective. Now we finished the meiosis division and we saw the cancer of the nanotechnology, which was invented by Dr. Mustafa Sayyid on the cell division. Okay, the technology or the application on the cell division. We finished the mitosis and the meiosis. Mitosis is the division in the somatic cells or in the body cells, while meiosis is the division only in the reproductive cells. Okay. Mitosis is for growth and compensation of the damaged cells and for asexual reproduction, while meiosis is for the formation of gametes for sexual reproduction. Okay, this is very, very important. Inshallah, the second lesson of this unit, we will talk about sexual and asexual reproduction. Okay, see you next video, inshallah. Bye-bye.